All right, in this video, we're going to discuss the paper that you're going to be reading the entire 8 weeks long. And there's going to be two assignments that come from this paper. There's a set of questions that are going to be due at the same day that exam 1 is due, that we take exam 1. And there is going to be a second set of questions that will be due at the same time as exam 2, meaning at the totally end of the class. So together, these make up about 20% of your grade. Now, the paper that you will be reading is entitled UTX and JMJD3 are histone H3K27 demethylases involved in Hox gene regulation and development. There's a lot of stuff there, but I guarantee you folks, within the next couple weeks, you will understand what all of this means. And I'm going to help you here by discussing it a little bit. Now, this is a paper that was published in the Journal of Nature, perhaps one of the highest impact journals out there. So it was a very exciting paper when it was released back in 2007. And one of the reasons that I like this paper is that the techniques used are extremely valuable and extremely important for you to know. And so the techniques that you'll learn as you learn how they did the different experiments in this paper are going to be very important. But instead of going into those, the purpose of this video is to simply show you essentially what this title means. What is an H3K27 demethylase? What is that? Okay. Now, there are several places that you can find to help with understanding this. And the first is in the primary paper alone. If you go all the way to the last figure here. This figure shows you the model of what they think H3K27 demethylase, so in this particular figure, K27DM, is that enzyme that they're interested in, this demethylase. And what this represents here is DNA is the black that is being wound around that histone octamer. Remember, 2 copies of histone H2A, H2B, H3, and H4. Well, we're interested in H3. That's what it means when they say H3K27. That means histone H3 and the 27th amino acid of that histone H3, which is a K, which is a lysine. Because remember that each one of the 4 different types of histones that make up that octamer have those N-terminal tails that stick out from the octamer. And that's what you're seeing here. You're seeing the N-terminal tail of H3. And the 27th amino acid of that is a lysine, K. So that's H3, meaning histone H3, K27, meaning the 27th amino acid, which is a lysine. And then trimethylate, Me3 means it's trimethylated, means it has been modified to have 3 methyl groups off of that amino acid. And that's what these 3 red circles are, trimethylated. Now remember that the modification of these N-terminal tails of the histones can do one of 2 things. They can, and oftentimes they'd end up doing both at the same time. They regulate just how compact the chromatin is, and they recruit specific proteins. So when H3K27 is trimethylated, you see 2 things. Number 1, you see that the chromatin is packed relatively tightly. And then the other thing you see is there's a particular set of proteins that are attracted to this trimethylation that between the 2 of them, between this packed chromatin and these proteins, this gene here that it can either be transcribed or be turned off is in fact turned off. Now, these 3 methyl groups can be removed. Okay, they are covalently bound, but it's an enzymatic reaction will remove them. And this was the exciting part of this paper, is for a while, scientists thought that once you methylate one of these histones, done, that this gene will be permanently turned off because these groups are permanently methylated. But once they discovered our K27 demethylase, so UTX, JMJD3, those are the homologous proteins. Both of them are K27 demethylases, meaning they specifically demethylate K27 on histone H3. And with the activity of that, K27 becomes demethylated. At the same time, there's another protein that isn't really mentioned too much in this paper called MLL, which trimethylates a different amino acid within histone H3's N-terminal tail, and that's K4. So this is going to change what's called the epigenetic mark. We started with demethylated K4, trimethylated K27. Then in comes our 2 enzymes, MLL, K27DM. K27DM demethylates K27. MLL trimethylates K4. That change within the chromatin opens it up. Look at that. It's opened up. That recruits a new protein, right? These proteins, since these modifications have been reversed, these proteins don't bind there anymore. Instead, a new protein binds, an activator, which binds there, can access the promoter and can start gene expression. Okay. Now, these 2 genes that demethylated K27, these 2 proteins that demethylated K27, that methylated K4, they're part of a large complex called the trithorax complex. The proteins that are responsible for demethylating K4 and trimethylating K27, they're involved in something called a big protein complex called the polycomb repressive complex. So these 2 are sort of like battling complexes. One is trying to modify things to turn on the gene, and the other is trying to modify things to turn off the gene. Now, what gene is this paper looking at? The regulation of what gene? And that is the regulation of HOX, specifically HOXB1. Now, they're not necessarily interested in what the function of this HOX gene is. They just know that their gene of interest, the gene that encodes K27 demethylase, regulates the expression of this HOXB1. So make sure that when you are learning about the histones and when you're learning about the N-terminal tails, that you think about this, that you think about H3K27, this demethylase, and what it's doing and how it's changing gene expression by changing chromatin. JMJD3 and UTX are both H3K27 demethylases. They are homologs of each other. In some of experiments, you're going to see them using UTX, and in some experiments, you're going to see them using JMJD3. Please don't be confused by that. Now, make sure when you're reading this paper, when you're looking at the figures, that you read the figure legends. It's going to be very, very important. When you look at these figures here, A, B, C, D, they look very complex. You've got to make sure you read the figure legends. And I'm going to show you how to do that in lecture. And I'll make some videos that will help you with this as you start to read. I highly recommend that you, at the very least, even though we haven't gotten into the techniques behind this, download the review paper. So H3K27 demethylases at long last, right? This is a paper that did not do the science, but simply reviews the science that was done. It's only about 4 pages long, and it might give you a good feel for when you then get to reading the primary article itself.